Now I'm going to talk a little bit about Mendelian traits, but in this case, we're going to go beyond those Mendelian traits. So I know that you have studied Mendelian traits since high school, and I did the same. And to my surprise, when I went to university and then when I did my PhD, they showed me that inheritance patterns are often more complex than predicted by simple Mendelian genetics. And in this class, I'm going to show you that, and I hope that during all the semester, I'm going to show you that the relationship between genotype and phenotype is rarely as simple as shown in this example. Uh, and I'm going to show you many examples that there are many hierarchical, many characters that are not determined by one gene with two alleles. There are many examples of that. And possibly the most important is the last one, is that despite that, no matter how complicated are those patterns of inheritance, they still follow the principles of segregation and independent assortment. So in the same way that Einstein talked about quantics, we still need to understand gravitational rules proposed by Newton. In the same way, the old thing is still true, although we need new things. The same thing happened with genetics. Mendel is still applied to all the phenomena, although his theory need to be expanded. And I gave you a small introduction in the video lecture where I give you examples where there is no complete dominance or genes are not completely recessive. And I show you that in some cases there are genes that are controlled by multiple alleles. And I also show you that there are cases that there are some traits that are polygenic, so they require not one, two genes, they need many genes to work together. So now, in addition to these ones, I'm going to talk about another examples that go beyond this Mendelian inheritance. And I'm going to talk about penetrance and expressivity, pleiotropy, epistasis, and the environment. And next class, we will talk about some examples with the pedigrees, that these ones will be also in the test. So let's start with penetrance. Now I'm going to talk about expressivity and penetrance. And to do that, I'm going to start with expressivity. And in terms of expressivity, I want that you see this cross. Here you have a homozygous dominant, and here you have a heterozygous, a homozygous recessive. Okay, this is not true. So, and what we learned in the previous video lectures is that if you have purple and white with the flowers, the next generation in the heterozygous, you will find that all of them will be purple, similar to the homozygous dominant. Although they will be heterozygous, the dominant will prevail and all of them will look similar. However, if you see here, you have a variable expressivity. Some of them will look very similar to this one, and other ones will start looking more similar to the white one. It will be like a mix between both. Yeah, so you have variables, degrees of expressivity of the combination of these two genes. And this is pretty common. So this term, variable expressivity, refer to genes that are expressed to different degrees in different organisms. So remember, this is an exception of the Mendelian rules. There is here, instead of talking about dominant and recessive, although it still work, you can find that the phenotype can have different degrees. So instead of being all of them purple, here you will have purple in different degrees: light purple, dark purple, and intermediate purple, for example. Now we're going to talk about another phenomena, that is penetrance. And penetrance refers to the proportion of organisms whose phenotype matches the phenotype for a given trait. So let me show you what is this. So here you have two homozygous, and here what are you going to get? All of them will be the same. The simple example with the purebreds that we have in the study. However, this is called complete penetrance. Why? Because the phenotypes match their genotype. So here this will be 100% penetrance because you count 1, 2, 3, 4, 5, 6, 7, 8, and 9. So 9 out of 9 display this phenotype. However, this 100% of penetrance not always take place. So in some cases you can have a variable penetrance. So now here, for example, you have 5 out of 9 are expressing the phenotype. So you have around 50% of penetrance. So we just talk about these two phenomena, expressivity and penetrance. And the interesting thing about these two phenomena is that they can take place at the same time. So for example, I just talk about the variable expressivity. So you see all of them looking purple, but in a gradient. So not they are dominant. The dominant purple is expressed in all of them, but not in a similar degree. And I also show you an example of variable penetrance that some of them will look purple, but not all of them are going to express that gene. But in some cases, you can have both at the same time. So you can have a variable penetrance and a variable expressivity. So you can see the gradient plus some genes that are not expressed. Yeah. So in some cases, and why? The always I will tell you depend on the mechanism. How the genes and the proteins are forming that trait determine when there is a penetrance, when there is a complete penetrance, when there is variable penetrance, when there is variable expressivity. So we will have to focus in each gene and try to understand how this gene is turned into a phenotype. And I will show you some examples later. So we just talked about penetrance and expressivity. So this is done. Now we're going to start asking, okay, but what is pleiotropy? And to explain pleiotropy, I want to show you this schematic. Here you see one gene, and that gene is involved in the is expressed in eyes, in the liver, and also in the heart. So there is one gene affecting multiple traits. And here you find another schematic. In here you find one gene number 2 that is affecting 4 different characters. So what is pleiotropy is when one gene can affect affect more than one trait. So we don't have one gene and one phenotype. There is not this linear relationship. You can have one gene affected multiple traits. And the story is not as simple as that. Sometimes you can have not only that, but you have pleiotropy as well as the effect of the environment. And later we will talk about that. And so now that you remember this, that you learn what is pleiotropy, now we're going to talk about the opposite, that is epistasis. Epistasis is when you have multiple genes interacting to form one single character. Okay, so let's talk about epistasis. So now we covered these two topics, and now you know more or less what is epistasis, and I'm going to show you more examples. So I want to show you this example of a dihybrid cross, and here you have your F1 and F2. And you remember this 9:3:3:1 ratio? When you have epistasis, you can have a modification of this ratio. So for example, so you can get different ratios of that. And why? Because there is gene interaction and because one gene can be masking the expression of another gene. So remember, epistasis refers to any type of gene interaction that results in a F2 dihybrid ratio that is modified from this 9331 because one gene is masking the expression of the other gene. And this is an example. Here, instead of getting a 9331, you have 97. So you get still 16. Here you still get 16. So these three here, let me have it in blue. This now becomes 7 because there is a masking of a gene. And there are multiple examples of that. Okay, to finish the topic of epistasis, I just want to remind you, if you see the last example here we have 9, 3, 3, 3, 1, but each column here represents a different phenotype. So you have a 9, 3, 3, 1 here on the top. However, here you only have at the end one phenotype, two phenotypes. So this 331 is the one that formed the 7, as I said before. So in the test, I might show you this table, and I want that you tell me what is that. Is this an example of what? Pleiotropy, epistasis, codominance. What is this table showing you? If you want to know more about epistasis, in genetics, we're going to do some, in a future course, we're going to give you some examples and some exercises about epistasis. But for this class, there is no need that we solve complicated exercises, questions about epistasis. I just know that you tell me that what is epistasis, and that you know that there are 9 possible dihybrid ratios that are included here. And here, as I said before, that you remember that each one of those rows represent a different phenotype. So we have talked about maybe things that you have heard or maybe that you never heard before. There are penetrance and expressivity, pleiotropy, and epistasis. And now we're going to talk about the environment. However, I'm going to give you a summary of the interaction of all these three processes. And that I already talked about that in a previous video lecture, is that, as I said in the beginning of this class, the relationship between genotype and phenotype, although in the beginning, when we start studying genetics, we thought that we're going to have one gene and we're going to have one trait, and then a new gene appear and you will have a second trait. So you will have one gene for blue eyes, there will be one gene for brown eyes, there will be one gene for green eyes, and this is how traits appear. This seems to be very different. Yeah, what we have is not a simple relationship between genotype and phenotype as we were expecting here. Yeah, this only takes place in very few cases. What we generally have is a complex interaction between genotype and phenotype. And here I show you that one gene can be affecting multiple traits, and one trait can be affected by multiple genes. So that they could be epistasis, and they could be a pleiotropy. So one trait can depend on multiple genes, and also within that they could be different gene expression. So for example, if you see the intensity of each one of the lines is different because each gene is expressed at different time and in a different degree. So spatial and temporal patterns seems to be very important. So instead of having thousands and thousands and thousands of genes, we're expecting in this model to have 100,000 genes in the 90s. And what we found with the genome project is that there were around 20,000, similar to fruit flies, very close to fruit flies. And why we have so few genes? because we have a complex relationship between genotype and phenotype, and we have one gene involved in multiple traits, and we have multiple traits involved in multiple genes, and gene expression is very important. Yeah, so in some cases you have if you express the gene in a higher degree, it could produce a very different phenotype. If you express it at a specific region and at a specific time, you will get a different phenotype. So all of this is that you remember that this simple relationship between genotype and phenotype doesn't exist in most of the cases, only in few of them, and most of them will show a complex relationship between genotype and phenotype. Okay, so now that we study all those things, we're going to finish in this class with the environment. That is possibly one of the topics that we need to study in more detail, and that we have been studying also for decades, but there is just a lot of things to understand from the environment. So previously in a video lecture I have shown you this relationship between genotypes and phenotypes. Now we know that over the last decades, every time that we talk about the environment, the influence of the environment is becoming bigger and bigger. We are not saying that the genes are not important. We just said that if you remember Galton, with Galton, the influence of the environment was almost zero. Yeah, it was almost not important at all. What it was more important, it was the idea of the genes, and this idea has prevailed, has been spread for many decades in biology, in genetics, and now every decade we say, okay, the mechanism is very important, and to understand the mechanism, the environment is very important. And now we know that most complex traits are affected by not only multiple genetic but also environmental factors. And we watch a video in previous lectures that talk about that there are very few genetic disorders that are involve only one gene. Actually, many of them involve several genes at the same time, and also they involve the environment. So in the future, and we are trying to understand that, what is the influence of both genes and environment? And I'm going to show you a few examples. So one trait that I found fascinating is the first one that I'm going to talk about, and I'm going to show you a video, is why human height has been increasing. And to understand that, we need to understand that this is a polygenic trait. So instead of having one gene that determines that you are tall or short, we have multiple genes affecting that trait. And we have examples of epistasis and Pleiotropy. This gene and this trait seems to be very complex, and the environment seems to play a key role. So, for example, it has been shown that if immigrants, families that come to a country that has food available, the new generation that grows in that country, it will become taller. And also another factor that had also played a role is for centuries, as now we live in cities and we have water available and we can wash our hands and we don't have to spend all our life fighting with diseases. Also, this seems to have influenced a lot, increasing the height in humans. So let me show you a video that they discuss about that.
These days, the average American woman is about 5'4", while the average Joe American male is about 5'9". But human height has had its ups and downs over the centuries. 3 million years ago, our ancestor Australopithecus only stood about 4 feet tall. 1.5 million years later, Homo erectus, the first early human to use complex tools, reached up to 5'7". And by the Stone Age, men of the Gravettian hunter-gatherer culture in Europe stood an average of 6 feet. Most of the historical data we have is for male height because reasons. Then agriculture happened. When Europeans switched to a lower protein, higher grain diet, men gradually lost 8 inches in height on average. And they stayed that way for thousands of years. By the time the 18th century rolled around, the average European man was only 5'5" tall. But when those Europeans emigrated to America, their kids grew up to be 5'8" tall on average. A huge jump in just a generation. During the Industrial Revolution, heights took a dip due to urban crowding and disease, but soon after, the human height boom continued and continues today. Every decade for the past couple centuries, Europeans have grown an average of about half an inch. Today, Dutchmen are the tallest people in the world with an average height of just over 6 feet. Back to where those Gravettians started 8 millennia ago, and almost as tall as me. These fluctuations of height, sometimes within a single generation, show that our environment determines a big part of how tall we are. But people in different regions and different families show us that height has genetic causes too. So which has a bigger role, nature or nurture?
Okay, so here they talk more about this debate. I'm not going to show you the entire video. We still don't know what is more important and about if it's nature, the genes, or the environment, but we start finding the different mechanisms that are involved in determining height.
These days, the
So one example that explains the relationship between gene and environments is to study fruit flies. And although nature and nurture is very important, it's very difficult to study in humans, and in some cases it can be unethical. However, using fruit flies, we can understand in more detail what is happening. So let me show you some examples. So I'm going to start showing you 3 examples: temperature, crowding, and then I go to masturbation. So we know that when you have normal flies, the wings will be straight. However, with one mutation, you can have these curly wings, and the flies, they will start jumping because they cannot fly. This is not functional. So when you have 25 or 25 or higher in temperature, the flies will be like that when you have this mutation. However, if you grow the flies lower than 20 degrees, the flies will look completely normal, although they have that mutation. So they are still curly, they have this mutation, but they don't express the gene. So this is an example that shows that the environment and the genes are interacting, and in this case, the phenotype will look completely the same to the wild type at a lower temperature. Okay, now we're going to talk about crowding, and to talk about crowding, I think if you work with fruit flies, you know that here is the food and the flies grow very happy here in this vial. And you generally have to have between 20 and 40 in these small vials. However, from time to time you have so much work that you might forget that the flies are here, so they end up having a lot of more flies there. They have many kids, many baby flies, and now you have 10, you can have much more flies. And when you study those flies, then in normal conditions, they will have this body size. But if you go and study in crowded conditions, the flies will become proportionally in all these body parts will be smaller. So you will not have small wings or small legs, but big head. You will all the fly will become smaller, and this is interesting because to regulate this phenomena that is called allometry, that you have this symmetrical disproportional reduction or increase in size, the same genes that are involved in these allometric changes, when you modify them, uh, those those genes are involved in cancer. Why? Because it seems that the way that you control the growth is very, if you misregulate it, you can lead to asymmetrical changes that it can lead to the cancer progression. So something that it seems to be not important, like how many flies you have in a vial, is very important for studying human diseases. And something similar happened with humans. If you don't have enough food, you will people is not going to be as tall. And okay, so to finish, so we talk about these four examples that go beyond Mendelian inheritance, and now we are going to solve some exercises. Next class, we're going to talk about pedigrees, something that you already studied in high school and is in the Campbell biology, and that it will be that it will that we will, there will be some exercises in the test. So you can read it and try to remember those topics.